What's up, Microbiology Nerd Family? Welcome to another episode. Today, I'm going to be introducing you to Staphylococcus aureus. Let me start with something that surprises people. We can find Staph aureus in two patients on the same day. One patient gets sent home, while the other one ends up on IV antibiotics. Same bacteria, different clinical significance. So today, I'm not just introducing you to Staph aureus. I'm going to show you why this bacterium is harmless in one place and dangerous in another, and how we decide that in the lab. Staph aureus is actually part of the normal flora for many people. It commonly lives on our skin, in our nose, especially the anterior nares, which is just a fancy way of saying nostrils, and sometimes in the throat or perineum. Around 30% of healthy people carry Staph aureus, often without symptoms. This is called colonisation, where bacteria are present and growing without causing you harm. Infection occurs when microbes invade tissues and cause damage, leading to an immune response and symptoms. Colonization does not always mean infection. Just because we find Staph aureus doesn't automatically mean disease. In the lab, context is everything. Staph aureus from a nasal swab and Staph aureus from a blood culture are two very different conversations. While it often lives harmlessly on our bodies, it is an opportunistic pathogen, meaning it will cause infections if the conditions are right, such as a breach in the skin or a compromised immune system. So what happens when a sample arrives in the lab? The first steps we take depend entirely on the sample type. Not all samples carry the same clinical weight. A skin swab, for example, will be cultured onto agar plates, but a gram stain isn't routinely performed. A blood culture, on the other hand, will require immediate gram staining and culture. This is a critical sample because the result has direct clinical consequences and can change patient management within hours. Let's say we have a blood culture sample with clinical details stating sepsis. At this stage, we want to know if the sample has any growth of potential pathogens, so we start with a gram stain. Under the microscope, Staph aureus appears as purple gram-positive cocci in clusters. The reason Staph aureus stains purple relates to its cell wall structure. It possesses a thick peptidoglycan layer in its cell wall that retains the crystal violet dye used in the gram staining procedure. Fun fact, on culture, Staph aureus often forms smooth round colonies with a golden yellow pigment. Staphylo comes from the Greek word staphylae, which translates to bunch of grapes, while aureus is the Latin word for golden. So the name Staphylococcus aureus literally means golden grape clusters. The gold comes from a pigment called staphyloxanthin. I'll let you in on a secret. Not all Staph aureus looks golden on a plate, despite the name. Pigment expression varies by strain and growth conditions. Some appear cream or white on culture plates. Staphyloxanthin is a carotenoid pigment that acts as a virulence factor by shielding Staph aureus from the host immune system attacks. So when you see golden cultures, you know this is one badass bacteria able to protect itself from the immune system. Going back to our gram stain, it cannot distinguish between species that share the same gram reaction and morphology. For instance, Staph aureus looks similar to Staph epidermidis on a gram stain. So although useful, a gram alone can't help us identify Staph aureus to its species level. So we use further biochemical tests to help us identify it. Next, we perform a catalase test. A catalase test detects the enzyme catalase, which breaks down hydrogen peroxide into water and oxygen. A positive test results in the production of bubbles. Staphylococci are catalase positive, which distinguishes them from streptococci and enterococci. Once we know we're dealing with a staphylococcus, the next key step is determining whether it's coagulase positive or coagulase negative. The coagulase test is then used to differentiate coagulase positive strains from coagulase negative staphylococci. It detects the enzyme coagulase, which converts fibrinogen in plasma into insoluble fibrin, causing visible clumping or clotting. The coagulase test is central to identifying Staph aureus. Most clinically significant strains of Staph aureus are coagulase positive. However, it's not the only coagulase positive species. Staphylococcus argenteus is also coagulase positive and very closely related to Staph aureus, although they are genetically distinct species. That's why we don't rely on a single test. Identification is based on a combination of morphology, biochemistry, and sometimes molecular methods. In routine practice, though, a coagulase positive Staphylococcus is most commonly Staph aureus. Once all biochemical tests are performed, we culture the sample onto agar plates that help facilitate its growth. On blood agar, it often grows as golden colonies, and on mannitol salt agar, it can ferment mannitol, turning the agar yellow. Here's something interesting. This is a McConkey plate. It's selected because it contains bile salts and crystal violet that suppress most gram-positives. Both these isolates growing are Staph aureus. It's fascinating because it's not supposed to grow very well in this media, which is designed more for enterobacteriales. But here it's growing in different colors. We often teach organisms as fixed personalities, when in reality, they show us that that is not the case, which to me is quite fascinating. So why is it considered pathogenic? Staph aureus is highly pathogenic because it produces multiple virulence factors. These include protein A, which interferes with immune recognition, coagulase, which helps evade phagocytosis, cytotoxins, which damage host cells, exfoliative toxins leading to scalded skin syndrome, and enterotoxins causing rapid food poisoning. This allows it to cause skin and soft tissue infections, toxin-mediated disease, invasive infections, and few bacteria have such a broad clinical spectrum. Clinically, Staph aureus is associated with abscesses and cellulitis, post-surgical wound infections, bacteremia and sepsis, endocarditis, osteomyelitis, pneumonia, septic arthritis. It is also a major cause of healthcare associated infections. Staph aureus is exceptionally well adapted to hospital conditions because it tolerates high salt concentrations and survives well on dry surfaces. It can persist on hospital equipment, bed rails, and skin. It's also why it grows on mannitol salt agar, which inhibits many other bacteria. Because of this, accurate identification and interpretation are essential. It also matters because of antibiotic resistance, including MRSA, which we'll talk about in a separate video. In the next videos, we'll look more closely at how Staph aureus causes specific diseases and how we diagnose them in the laboratory. But before you go, let's try a quick micro quiz to see what you picked up from this video. Take a moment to think about the answers, and if you'd like, share your answers in the comments. I'll go through the correct answers in the next video. I'll start off with an easy one. Question 1. About what percentage of healthy people carry Staph aureus without symptoms? Question 2. What feature of the bacterial cell wall allows Staph aureus to retain the crystal violet dye during gram staining? Question 3. Now this one's slightly tougher, but I know you guys are going to manage. What is the name of the carotenoid pigment that gives some Staph aureus colonies their golden color? Question 4. The catalase test distinguishes Staphylococci from which other group of gram-positive bacteria? Question 5. Which selective medium allows Staph aureus to grow because it tolerates high salt concentrations? Okay, final question. If a gram stain from a blood culture shows gram-positive cocci in clusters, does that confirm the patient has Staph aureus bacteremia? Now with this one, the answer requires not only a yes or no, but also the reasoning behind your answer. Feel free to pause the video if you'd like time to think through the answers. That's all for today, folks. Please make sure to like, comment, and subscribe. Stay curious, and I'll see you in the next video.